I'm going to start out and introduce my very good friend and colleague, collaborator, Dan Lowenstein, who is the professor and vice chair of neurology at UCSF, right around the corner, who is going to talk to us about new directions in epilepsy research and get us really excited about what's coming down the pike.
Thanks, Jackie, and good morning. And I'm going to launch right in to make sure that we stay on time. So Jackie gave me what struck me as a pretty tall order about 6 months ago. She said, basically, review all of the exciting things in epilepsy research in 30 minutes. So that's what I'm going to try to do. Prepare yourself. 6 quick stories, an average, I think, of 5 minutes each. And here's what they are. I'm going to talk about Seizure Laser Tag, Genomics Rules, the ultimate model system, U, fishing for drugs, interneurons to the rescue, and thinking big about a project, the Human Epilepsy Project. My apologies to the bench scientists in the room, as well as the pure lay audience. I've done my best to try to come up with something of a balance to make each of these topics understandable to some degree. Okay? You can tell me if that works in a moment. Okay, so let's talk about Seizure Laser Tag. This refers to a really spectacular report from John Huguenard and Jean Paz down at Stanford last year, in which they essentially showed that using a particular form of light on a particular type of channel can interrupt seizures in an in vivo model. So let me walk you through this. They used a very nice focal stroke model in which there's a thrombosis of the cerebral cortex in the animal. And John's group and others have known for a long time that there are important connections between the cortex and the thalamus. And the idea here was to see whether the thalamus, how it might be involved in a focal stroke model. And they demonstrated that was clearly the case. Here you see a control neuron versus one. This is a thalamic neuron in the injured animal. So even though the damage was out in the neocortex, they demonstrated that the neurons in the thalamus actually get injured, and they have abnormal activity. This is a control neuron in the thalamus, and this is an injured one showing spontaneous high-frequency spiking activity. So they asked the question, is there a way that we might be able to modify the activity of these thalamic neurons and alter the epilepsy behavior in the animal? Now, to understand what they did, we need to go through very quickly exactly what's going on with optogenetics. So probably everyone's heard the term. Everyone knows it was one of the scientific breakthroughs over the last couple of years. It's certainly in neuroscience. But just to make sure that you understand how this works, here's the 6 steps that are involved. You need to actually get an opsin, which is a very special type of protein, which is sensitive to particular wavelengths of light. And you put that behind a promoter that will put the opsin in the proper cell that you're interested in. You can package that into a virus, inject it into the animal, and hopefully the opsin is going to find its target. Now, once you've done that, you need to have a particular type of light wave, in this case a laser light beam, which when it reaches that opsin will open up the channel and modify the behavior of that particular cell. Now, just to demonstrate what this looks like, the opsins actually are sort of universally found in nature. They're light-sensitive molecules, and in this case, this is of some flagellated organism. You take out the DNA, you insert it, as I just said, say with virus packaging. It finds its way into a particular cell type, and then that cell expresses the opsin along the surface of the cell, and because it modifies the flow of ions in or out of the cell, you can actually, in a very targeted way, change the behavior of that individual neuron. So once you've done that, then you can do a whole variety of experiments, physiology and so forth, on that modified cell type. So that's what John and his group did. They created the stroke in 1-month-old animals. They then placed an inhibitory opsin in those targeted to those thalamic neurons that we just saw. implanted the laser device and an EEG into the cortex, and then did chronic recordings and stimulation and monitored the animals. So that's the experimental setup. So here's what they found. So when the animals would have their spontaneous seizures, which was part of the stroke model, and they turned on the laser beam that was focused on those thalamic neurons that would cause inhibition of those abnormally excitable cells, the seizure stopped. And that's shown here. So here's the onset of the seizures by EEG, the laser, and then interruption of the seizure. And then they went on to actually create a closed loop system. Now that they demonstrated that turning on the laser could abort the seizures, well, what about creating a system where the EEG, when it picks up the onset of a seizure, automatically turns on the light? And that's what they did. So here's the Eeg Recording System. They have a processor that reads the seizure onset, it turns on the laser, and then they can determine whether or not it works, and they demonstrated that, in fact, it does. Here in this left-hand panel, you see the onset of the seizure. It's detected, the light's turned on, the seizure never actually occurs. That's compared to the detection with no light, in which the seizure does occur. So a beautiful demonstration of a very, very sophisticated, refined way of targeting individual neurons and with a closed-loop system aborting a seizure. Okay, second topic, genomics rules. So this is a really nice figure from Ingo Helbig, one of our colleagues in some work that's going on in Epi4K. This is a figure that he put together for a review that we published together, but this is all Ingo's ideas. So this is basically an overview of the history of epilepsy genetics. It all began in 1995 with the paper by Sam Berkovic and colleagues describing the first true epilepsy gene, a mutation in the nicotinic acetylcholine receptor. And as you can see, this was discovered in part because of a very informative, remarkable family in which epilepsy clearly runs through the family. And so with that discovery over the next number of years, most of the epilepsy genes that were found, in fact, turned out to be ion channels. So we could refer, Ingo refers to that time, up until 2000, as the channelopathy era. Well, then we had about actually over 100 candidate gene studies that essentially all negative results. So this was using genome-wide association studies with, unfortunately, not sufficiently large enough numbers of patients. And so Ingo refers to that as the Dark Ages. In more recent years, we've had a whole host of studies that have been looking at so-called copy number variants in the genome. So these are variations in our genomes which are either additions or deletions or duplications. And some very important work from a variety of investigators around the world have demonstrated that these so-called CNVs are in fact important, and so we can refer to that as the CNV revolution. But really where we're at now, because of the extraordinary advances that have been made with sequencing technology, we're really at the next generation sequencing bubble. Just one example is the work that came out of Epi4K that built on the work that was done by the Epilepsy Phenome Genome Project. And this is a paper describing de novo mutations in patients with epileptic encephalopathies. In fact, in this work, there were 264 individuals with either infantile spasms or Lennox-Gastaut syndrome that had exome sequencing along with their parents. That's 800 exomes. And in that work, a whole number of de novo mutations were discovered. Some were previously known, some were new, and in fact, there's even a longer list of de novo mutations that are likely causative in this population, but we really just need more numbers to be certain about that. The thinking here is at this point, we probably can explain about 15% of the genetic basis of all epileptic encephalopathies that don't have an obvious, say, structural or metabolic cause. Now, I know that Steve White gave you an overview of a number of things going on, and he mentioned the Epilepsy Genetics Initiative. I just wanted to touch on that a little bit more because it really, this is a fantastic idea that really David Goldstein came up with at Duke, and that's been embraced by those of us in Epi4K and now and now cure. And the idea here is that the clinical sequencing that is being sent more and more and more to the companies out there is really relying on the in-house understanding of an individual company. So that the company is relying on the information that they get from the literature or their own analyses. But really, we're in the era of epilepsy genomics, not just epilepsy genetics, genomics. We need as many genomes as we possibly can to really understand what the landscape is for all forms of epilepsy. So the idea here is, why not create a data repository that includes not only the exomes and the genomes that are being produced in research labs, such as the work going on in Epi4K, but also brings together the information that's being generated by a growing number of clinical testing labs. So this really is the basis of the idea of the Epilepsy Genetics Initiative, and I'm going to just show you a video.
Imagine having the ability to unlock the genetic secrets of epilepsy. Just think of what this would mean for the 65 million people currently living with this debilitating disease and the doctors who struggle to find a way to stop their seizures. Knowing what causes epilepsy and what treatment might work best seems almost like an unattainable dream. But thanks to modern advances in genetic research, this dream is close to becoming a reality. By examining the changes in a person's genes, researchers have been able to identify altered genes that lead to some people's epilepsy. With about 20,000 genes in each of us, this is no easy task. However, by using genetics to diagnose and treat each person individually, we could create a world where we tailor treatments to each individual person with epilepsy. Unfortunately, current gene testing is limited because studying gene data is challenging, and analysis sometimes needs to be repeated again and again over time before we can identify a cause. Now, that's where we come in. The Epilepsy Genetics Initiative, or EGI, is a project of CURE dedicated to advancing epilepsy genetics. CURE has created a place where people with epilepsy, with the help of their doctors, can house their genetic data to have it analyzed and reanalyzed to help find the cause of their own epilepsy and help advance science as it seeks to uncover new causes of epilepsy that have yet to be discovered. Researchers will be able to study the genes which will be confidentially collected in EGI and build a database that has never existed before. Just imagine what this will mean for epilepsy research, therapy development, determining which treatments work best for which people, and discovering why some people with epilepsy suffer from coexisting conditions like depression and learning and memory challenges. By bringing together people with epilepsy, medical professionals, and scientists, EGI will help to improve the diagnosis and treatment of epilepsy in a way that has never been done before. Find out more at www.cureepilepsy.org/EGI.
So as we were developing this idea, one of my concerns was that the companies might not be that interested or willing to deposit such information into a common repository. And in fact, we've discovered just the opposite. And so this has been a beautiful experience, actually, in collaboration across all domains. Just a couple of other details about this. The idea now is, with a repository that's accumulating thousands and thousands of exomes and eventually genomes over the next number of years, that groups can reanalyze the data on some regular basis so that we can take advantage of essentially the numbers, because it's all in the numbers. Those results will be communicated back to the referring physician and their patients, and then the data will be made available to all qualified groups. So whether it's an academic group, a company, other investigators, that's what the repository is meant for. And finally, before I go to the next topic, I just want to mention Ingo, who I referred to just a couple of minutes ago, has been a remarkable blogger. And the International League Against Epilepsy, the Genetics Commission, has actually adopted his blog. And if you really want to go to the source of all exciting news related to epilepsy genetics, go to epilepsygenetics.net, and I'm sure you're going to be entertained. Okay, next topic, the ultimate model system, U. So everyone's heard about inducible pluripotential stem cells. I thought I would just walk us through more precisely what that means. So the idea here is that you have an individual, in this case the patient or ourselves, and by taking a skin biopsy, literally a biopsy of skin from the forearm, you can dissociate those cells, grow out fibroblasts, and with specific molecules, you can drive those skin cells to be inducible pluripotential stem cells. They're stem cells, meaning that they have the capacity now to go from their very embryonic, if you will, condition to be induced into different cell fates, to be made into essentially any of the cell types in one's body. When you have that type of iPS cell, or inducible pluripotential stem cells, there's two big directions you can go with that. First, you can actually, in the case of a patient who has a gene mutation, you can modify that mutation, you can correct it in the iPS cell, then drive it back to a healthy cell state, and theoretically transplant it back into the patient. And this is something that has already been done with blood-borne oncologic disorders. On the other hand, you can take these iPSC cells and drive them into a differentiated state, into a certain cell fate, and then use that cell type to screen for compounds that might be useful in a particular disease state, and then find disease-specific drugs and then try it in the patient. So that's the general idea of the power of iPSC cells. And many of you have, I'm sure most of you have heard of the fantastic work that's been going on in this area, led in large part by Jack Parrott and his colleagues at the University of Michigan. So Jack had the brilliant idea of deriving these iPSC cells from patients with Dravet syndrome, knowing, of course, that in many of those patients, the gene mutation is in the sodium channel subunit. And by creating that cell type from an individual patient, not only did you have the specific mutation, but you had it on the specific and truly unique genetic background of that patient, which is very important. And by using, again, specific factors to drive the fate of those cells, he could make, for example, as shown in this slide, neurons. Now, by studying those neurons, he and others have been able to show that there are clearly abnormal characteristics of the firing properties of neurons derived from patients with Dravet. That's not a big surprise, but this was a very clear, elegant demonstration of that, and it's actually made significant progress to understanding the underlying biology of Dravet syndrome. And as Jack is fond of saying, this is really human seizures, and this is like a human's cells in a dish, which is pretty amazing. Now, Jack also had the very clever idea of asking, well, what about the association between Dravet syndrome and SUDEP, which of course is, clearly one of the one of many tragic aspects of this form of epilepsy and epilepsy in general. And knowing that these various disorders of ion channels can predispose to SUDEP, they asked the question, well, what's the connection between the ion channel mutation in Dravet and the behavior of heart cells in those patients? So again, step back for a moment. He has iPSC cells. They can be driven to whatever fate one's capable of, so why not drive them instead of to neurons into cardiac myocytes? And that's in fact what they did. This is an example of what they look like. And by creating these myocytes, they could study them in the dish. And here are some videos of the behavior of these cells. So in the upper left-hand panel, these are control stem cells. And they grow, they differentiate, they replicate, and actually they create a syncytium. They actually create essentially a cardiac tissue in the dish, and they beat. And if you just watch the behavior of these cells, that is a beating piece of heart tissue. Now let's compare that to three cultures of myocytes taken from three different patients with Dravet syndrome. Each of them has a different mutation, although they all involve the sodium channel subunit. And the question is, are any of them associated with abnormal behavior of the cardiac cells? And I'll show you all three, and you can take a look and tell me whether any of them look abnormal. Here we go. There we go. So if you imagine taking the pulse of those additional cultures, any of them look abnormal? Actually, one of them looks abnormal, and it's the upper right-hand panel. It's actually having an arrhythmia. And so here's a really dramatic demonstration of the power of iPSC cells in being able to advance our understanding of the phenotype here related to cardiac function in Dravet patients. Okay, here we go again. Next topic, fishing for drugs. So this is a spectacular paper that came out of Scott Barabant. Scott, you're here somewhere? Yes, there he is. Okay, so many of you have probably heard about this. This is arguably one of the most important papers over the last year and a half to two. And this is a remarkable study in which the zebrafish was used as a model system for screening for drugs. So you probably have seen pictures of zebrafish. The work that Scott did actually involved this big guy here, although he's really little. This is the larva of this. This is why they're called zebrafish. They've got these stripes. That's the larva. And what Scott and his group have done is they've been able to, because these organisms are so relatively easy to manipulate genetically, they actually placed an SCN1A, a Dravet mutation, into these zebrafish and created essentially a Dravet zebrafish. And you can study these actually in little tiny wells, 96 well plates, and you can watch the behavior of the fish. And if you take a look in the upper right-hand corner, you're going to see the control animal here and a Dravet mutant here. And you'll notice that there's a heck of a lot more behavior going on the right-hand side compared to the left. That control animal is alive, by the way. You'll see it move every once in a while. But the mutated zebrafish is actually having seizures. And this was, you can hold these fish in place and actually do extracellular recordings and observe directly the seizure activity coming from this animal's brain. And the beauty of this system is that because you can study one fish per well, using a tracking system, you can, in an automated way, you could actually compare the behavior, the swimming behavior of, in this case, control fish versus those that had the mutation. And so Scott used this system to first test whether known anti-epileptic drugs have an impact on this model, and in fact, they do in a selective way and in a predictable way. So that was a proof of principle that the system actually could be effective in assaying for the ability of different compounds to control the behavior of the animals. And then they carried out a screen of a library of molecules, and here's the readout. And you'll notice that the y-axis is velocity, percent change from baseline. So using the seizure model, what we want to see is a decrease in the movement of the animals. And so anything that's down low is a possible hit. Of course, if it's too low right at the baseline, that means the animal's not moving at all, which means it could be dead. So you have to screen for that. But in fact, the red dots show a significant decrease from baseline of compounds that were tolerated by the animal. And lo and behold, the drug that really stood out was clemizole. Clemizole was a drug that was approved by the FDA in the mid part of the 20th century. It's an antihistamine that was planned to be used as a anti-bronchospasm agent. It never really got regular use. Scott, you probably know the details as to why, but he is really moving forward now to see whether this could be brought to use in humans. When he tested clemizole in the individual fish, he discovered the following: that is, the untreated animals would have seizures. Clemizole aborted them essentially completely, and this was not due to just paralyzing the animals, because using a drug that was a neuromuscular blocker, in fact, did not have an effect on the seizure activity. So really a fantastic discovery. Okay, interneurons to the rescue. Actually, this is another Barabant lab work. So here the idea is, could you come up with a way of just placing cells directly into the brain, into an epileptic region. The idea is you identify where the zone is, where the seizures are coming from. Then you transplant the cells into that area, hope that they integrate and control that focal area of network dysfunction. Well, the question is, which cells? I mean, the brain is a complicated thing. Here's a cross-section of the brain, and there's 10 to the 10th cells there to choose from. How do you come up with a cell that's going to be of benefit? Well, everyone knows that, for example, the neocortex not only does it have a lot of cells, it has many, many, many different cell types, some of which are excitatory, some of which are inhibitory. Probably it would be ideal to, in the case of epilepsy, to come up with purified inhibitory cells that would make sense for quieting an excitatory network. And in fact, a lot has been discovered over the last decade and more about how it is that the inhibitory interneurons make their way into the mature neocortex. And in fact, many of them come from a region deep inside the developing brain called the ganglionic eminence. The ganglionic eminence gives rise to these primordial cells that are destined to travel throughout the neocortex, find their location, and develop into inhibitory interneurons. So Scott and colleagues took advantage of this understanding of basic fundamental developmental neurobiology and asked the question, could we isolate the source of these inhibitory neurons and see if they develop in a way that we could transplant them into a seizure focus? And that's, in fact, what they accomplished. So here's what they did. They took the medial ganglionic eminence, which is the source of GABAergic interneurons, and tested whether or not they could migrate into a host brain. And then once they established that, tested whether or not they actually have mature firing properties and whether they integrate into the host circuitry. And the lower panel is part of the work that they did to demonstrate those last two properties. In fact, the cells do migrate, they do have mature firing properties, and they do integrate. This is what they look like if you take these medial ganglionic eminence cells and transplant them into the hippocampus. In the case of a very young animal, they'll migrate up to 5 millimeters. In a P60 animal, which is a very mature rodent, they'll actually migrate 2 millimeters. And these can migrate in the cortex, in the hippocampus, and the amygdala. So with this setup now, Scott and his colleagues had the ability to take isolated inhibitory neurons and place them into a seizure focus. And this is the work that they did. When they placed it into a genetic form of epilepsy, in this case, a potassium channel mutant, which had spontaneous seizures, and injected them at P2, they saw a 96% seizure reduction. This is in an animal that has a general dysfunction of the brain, of the cerebral cortex, due to a potassium channel mutation. But I think even more spectacular was using these cells in an acquired form of epilepsy. So they used the pilocarpine model of temporal lobe epilepsy. They did an adult transplant. Remember, that's at P60, where they only migrate a couple of millimeters. And they, in fact, found a 92% seizure reduction, specifically when the cells were transplanted into the hippocampus, not, for example, into the amygdala. And some of the comorbidities that are associated with this model were also reversed. Okay, last topic, and this one's really quick. Let's think big, the Human Epilepsy Project. This is a project that Jackie, Ruben Kuznecki, and a whole number of other people around the country and the world are working on. We had this idea about, God knows what, Jackie, 4 years ago. So this is a project that we believe deep in our hearts and our minds is really, really important. So important that even though we had some difficulty getting funding from NINDS at the beginning, I should have said at the beginning of my talk, actually everything that I presented couldn't have happened without NINDS. All the investigators that I've mentioned have been supported in major ways from funding from the NIH. With the Human Epilepsy Project, we really turn to industry supporters to help get us going, and NIH actually has been really helpful in giving us ideas as to how to continue this work. But the basic idea of the Human Epilepsy Project is we need to do something about the fact that we do not have any bioassay, we do not have any biomarker for disease activity in people with epilepsy. We have patients who come and want to know whether their epilepsy is getting worse, whether it's stable, or whether it's gone away, and we essentially have no way of knowing that answer. And until we do, the suffering that is associated with epilepsy and its comorbidities is never going to go away. So the Human Epilepsy Project is basically that, it's built around that whole idea. And so what we're doing, we've created a prospective observational cohort study. We're attempting to enroll between 500 and 600 people with recently diagnosed focal epilepsy. And this is a prospective study, so they're going to be followed for a minimum of 3 years. And what we're doing is collecting a lot of information about these individuals, both in terms of their clinical semiology, their imaging characteristics, their EEG characteristics, blood for DNA and serum for proteomics, and urine for metabolomics, as well as really detailed information on neuropsychiatric comorbidities. And the idea is that by having enough patients over time and following them carefully, we will indeed be able to discover the biomarkers that we so sorely need. So the real goal is to create an open repository of detailed clinical information and these samples for future studies. We need help, by the way, for this. We're underfunded, so if there's anyone out there who's interested in participating in this great endeavor, I can see Jackie smiling, I'm sure. We'll crowdfund it, there you go. So there you go, that was a quick foray. I tried to highlight Seizure Laser Tag, the ability to target individual neurons and stop seizures. We're in the era of epilepsy genomics, and the Epilepsy Genetics Initiative is a cool idea to expand that. I've told you about inducible pluripotential stem cells and the insights they're providing, the amazing zebrafish, transplanting interneurons into targeted areas and aborting focal seizures, and finally, the Human Epilepsy Project. So thanks so much for your attention.
This has been an incredibly wild ride through science, and because Dan isn't going to be here for that long, he has to go back and work on these amazing things. If there are any questions, we can take them now. He will be here for the rest of the morning, but it was that clear. Okay, so thank you very much, Dan. That was incredibly informative. Now I'm going to hand the mic back over to Roger Porter.
I'd be grateful if our panelists would come up on my right-hand side, please, so that the webcast works better. And I will introduce you as you are coming up. This is a terrific panel, one that I worked very hard, I have to say, to get together, and I'm very proud of it. It's the role of corporate and government partnerships in developing new epilepsy products. We have Santiago Arroyo from the Vice President and Head of Clinical Research and Chief Medical Officer of Pharma Therapeutics from Worldwide Research and Development from Pfizer. We have Roy Twyman, MD, Vice President, Global Head of CNS Development from Janssen. Both of these gentlemen are tasked with what does Big Pharma look for in a new drug for epilepsy. Then we have Dr. Michael Gold, Vice President, Global Therapeutic Area, CNS from UCB. What does a medium-sized epilepsy-oriented company look for a new drug for epilepsy? And finally, because drugs isn't everything, drugs aren't everything, we have Frank Fisher, the CEO of NeuroPace. What does the device industry look for when considering developing a new product in the epilepsy space? So I have invited each of you to have 5 or 10 minutes to tell us what you think right off the top before we initiate the actual panel discussion. And I suggest you come up here at the microphone, it'd probably be easier. So Santiago?
Sure, thank you very much, Roger. It's a pleasure to be here, and it's a pleasure to see so many known faces for so many years. So let me start, I think I have here this, by a disclaimer, of course. Everything that I'm going to say is my opinion, and just my opinion is not Pfizer's opinion. And I have a vested interest in Pfizer's well-being. So let me start with, when you think about what do we need for a new drug to be successful in big pharma, we need efficacy. We need a drug that is differentiated, and the best differentiation is efficacy. We want something that has superior efficacy that will really make a difference. There are other characteristics that we will look for, like having improved safety, tolerability, absence of dizziness, of somnolence, better delivery of effects, like no titration, better scheduling, and broader spectrum. And of course, it would be ideal to have a disease-modifying drug, although obviously we are very far from that. But that will make a real difference. Not only I think that efficacy is important, I think that mostly neurologists across the pond, I mean here and in Europe, believe that efficacy is the main thing. Safety appears as a secondary, tolerability appears as secondary, basically because many or most of our antipyretic drugs are already relatively well tolerated, and efficacy is the major feature that is missing. So this is a cumulative distribution plot, and for those that are less familiar with it, on the x-axis is the improvement measured by change in seizure frequency, percent change in seizure frequency from baseline, and you have on the left side, you have a reduction in seizures, on the right side, a worsening in seizures, and in the y-axis you have the percentage of patients. So, and this is one of the Lyrica pivotal trials. And you see the percent improvement, the 50% improvement, is about 45% or so. And this is very typical. The effects of pregabalin in this case are very typical of all of the major antiepileptic drugs. What we get is about 50% of patients improving about 40% of the time, and we have very few patients improving 100%, very few seizure-free patients. So what we want is to basically raise that line and have no seizure worsening, have a very substantial amount of patients that have a 50% response, and have many, many subjects, and I put here like 50%, that are seizure-free, because seizure freedom is what really makes a difference. So how can we do that? And there is not a clear answer to that, but I will think that it would be naive to think that one drug is going to be the magic bullet that is going to change epilepsy forever. Epilepsy is a very heterogeneous disorder, and no one drug will make that happen. So the only way to go around that is to try to understand who are the responders within the population of patients that you are going to treat. In order to get to the responders, you need a biomarker, and probably one of the best potential biomarkers is genetic variants. So how to get to that from analyzing common variants in an heterogeneous disorder is pretty complex, and there has been some efforts done in the past with 1,000 patients here, 500 patients there, 3,000 patients, but really you need tens of thousands of patients in order to get variants that will make a difference. And once you identify those variants, you can probably get to understanding their functionality and the directionality, and finally understand what will be the pharmacology of a potential druggable target. So we are far from there, but I am happy to see that there is already something in the epilepsy field that is starting, and hopefully we'll get to reach to those 20, 30, 40,000 patients that have good phenotyping and good genotyping done. The other possibility, instead of go to the common variants, is to go to the rare variants, to go to subjects or to patients that have a rare genetic disorder, and as Dan was saying before, get tissue, or in this case, blood from these patients, and do induce pluripotent embryonic cells, and from that differentiate those cells into neurons, and basically have the ability to analyze the properties of those neurons in a dish. Dan was saying before that you need a biopsy. There are some labs, our lab, that we can do that from blood. So you don't need anymore a skin biopsy. From blood, you can get to the iPS cell, you can get to the cortical neuron. So basically, today you have a screening platform that will allow us to understand the phenotypic information from that cell, the genomics information on the other side, and hopefully be able either to find a genomic marker or to understand the pharmacology of new targets. I'm happy to say that Pfizer, Intellimedics, and the Epilepsy Foundation of America are currently in the process of finalizing an agreement, and we will be working with children of Dravet Syndrome, trying to basically do what I have just mentioned here in this slide. So I'm very hopeful that we will make a lot of progress, not just for these children, but for many other people with epilepsy. So what is this is my last slide, by the way. Sorry for this. So what is the vision at the end of the day? I mean, where do we would like to be? I mean, something that is interesting to see is that many drugs for epilepsy work across different disorders. They work, valproic acid works in epilepsy, it works in migraine, it works in bipolar, pregabalin, the same. Many of our compounds work across different disorders. So would it be great, right? If there were some variants that were associated with the function of some channels or receptors that could be common across different disorders, and that will define those responders to specific compounds. Could we evolve like oncology has evolved? That right now, epidermal growth factor mutations are treated, not specific cancers. You treat the mutation, you don't treat the cancer. So if we were to reach that to that level, it would probably be very possible to have a high efficacy in a few patients across different disorders. I will be cost effective. Thank you.
Thank you very much.
A couple, a disclaimer or two also. My title's not in CNS development. We don't have a CNS development group anymore. Our organizations organize ourselves into disease areas, and I'm in fact in charge of the Alzheimer's disease area at Janssen. And that being said, so Janssen's entirely neurological focus is in Alzheimer's disease and neurodegenerative conditions, and epilepsy is not a priority. And so it's been a while since I've actually thought about epilepsy itself, so I've jotted some notes down here, and much of this actually follows our thinking about any opportunity as we get into potentially developing a drug product itself. And let me first start saying that the world of drug development and the pharmaceutical business of yesterday is not going to be the world of tomorrow. And so the industry is now challenged with what the future is going to be like. And that challenge is that we need to be innovative. And so the history of Me Too type drugs and things are not going to be really reimbursed substantially for large companies to be able to realize the profits and necessary return on investments from that. So the future of therapies tomorrow need to be innovative. And we heard a lot of fantastic things yesterday, actually, being innovative in the field of epilepsy. But when one thinks about the special timelines and challenges within the drug business, these timelines are quite long. So what's innovative today needs to be innovative when your product hits the market 5, maybe 10 years in the future. And in fact, it needs to be remaining innovative as it returns on the investment over the lifetime of that drug over the next 5 or 10 years as well. And so you need to be very, very forward-thinking in your innovation as going forward here. So the biggest troll, if you want to call that, is Father Time. Because Father Time keeps on pushing that innovation bar higher and higher and higher. And so we need to beat that innovation bar by the time this product reaches the market, and it also needs to be innovative for the life of that product itself. And so how do we think about that? So first and foremost is the unmet need, right? And can we access the market itself? And so clearly in epilepsy, there's an unmet need, and there is access to the market as well. And we all know, and we've heard this, is the refractory population needs better drugs, they need higher efficacious drugs. Epileptogenesis has been touched upon here. This would be a fantastic entry into this field as well. Santiago just talked about, and there's going to be some overlap between what I'll touch upon and what Santiago touched upon, is targeted medicine. This is the future. Is we're going to go after stratified populations and targeting therapies to specific patients with diagnoses or diagnostic approaches and biomarkers, as Dan Lowenstein pointed out. It's going to be really playing a big role in this in the future itself. And so this is another great opportunity in epilepsy. And also, there's the opportunity of improving the current therapies. We know that 60-70% of subject or patients are adequately treated with their drugs today, but the drugs are producing some interference in their lifestyle and things, and so there is an opportunity perhaps improving the overall risk-benefit profile itself. So that's number one, is the unmet need. The other one is that as we think about this innovation hurdle that we have, it has to be differentiable. The opportunity to differentiate must be there, and we must know that we have an opportunity to get to that. And this is a big challenge specifically for epilepsy, as we don't have really predictive mechanisms to identify, do we have a potentially useful drug that will truly differentiate in the future itself. And so this has been proven. This field has been extraordinarily successful in introducing new drugs for patients with epilepsy. There may be, 20, it's been a while since I've been in epilepsy, 20 drugs now. Can we show that any of these drugs are clearly differentiated from any of these other drugs? No. We have multiple mechanisms and things, but no clear differentiation. So we need to figure out how to bring forward new drugs that are truly differentiated, that beats that innovation hurdle, so one can get actually a return on investment in the future, as I pointed out. The other thing, the number 3 thing that we bring in is, where's the science? Is the science substantial? Is it really advancing? Will it provide us that innovation opportunity through the science? And so Dan gave a fantastic talk. This is really exciting, seeing where the science in epilepsy is going, because this is where a lot of the other fields have really taken the bull by the horns. They've moved away from animal models. Animal models have been good in sort of testing some pharmacology and things, but the animal disease models have been very inadequate. And then until we get the Ipsc Cells or other things coming forward here, animal models have not been all that useful except for some pharmacology itself. What we need in fields are things like molecular targets, and so Dan touched on some of those, and it's really exciting to see that perhaps the new wave of genetics, maybe copy number variations, things will add to this itself, and so this is extremely exciting to hear that for epilepsy. I'll give you an example. I said I was in the field of Alzheimer's now. The Alzheimer's has a couple, it's a proteinopathy basically. One of the key proteins, amyloid, has over 230 mutations pointing to amyloid as causal to the disease, which involves 2 gene targets there. 230 mutations establishing causality. And so we have a great molecular targets not only in amyloid, but also in the tau protein, where the science, and again, this exciting science says that there's something interesting about tau protein where it seems to be acting like a prime protein and spreading. So it provides a new target for pharmaceutical intervention. So that is an example of exciting science that drives industry into going after specific disease states itself. The other one is also in the sciences, can we translate it, or do we have translatable models? And this is one of the other things is that Dan pointed out, was again applauding the efforts here, and I've heard about some of the remote monitoring approaches from yesterday. Translatable models that allows us early decision making, such that we can actually identify what is potentially promising or what is potentially a dead dog and we need to kill it, early. So these early proof of concept approaches need to be established for epilepsy as well. And if there is a path for that, that makes it an attractive opportunity for investment into that type of disease area. The fourth thing is there a path for development? And so that's one of the positive things about epilepsy is there's a clear path for development for this. But we also heard some exciting news from Dr. Katz yesterday that perhaps there are new ways of thinking about enabling development in epilepsy itself. And this is actually going to potentially help pique the interest of industry with regard to development in epilepsy as well. So I applaud that quite a bit. And then the fifth area is what about the competitive landscape? In epilepsy, this is a problem. There's been a lot of drugs that's entered the market. There's a lot of generic drugs there. And so what does that mean from the standpoint of standard of care? The standard of care allows many treatment opportunities for patients where physicians have the choice of a multitude of drugs. And so the driving force, the economic driving force for this is that to try the cheaper drugs first, because there's really no difference with them. We haven't established differentiation, is to try the new drugs first before actually getting to a new product. And so unless the new product clearly has, as I said, that differentiation and providing that additional value, and it's going to be principally an efficacy value, you're going to really have a very difficult time competing for price in this highly genericized market itself. And then lastly is business opportunity. Is that, as we look at this long timelines for development, and like I said, it takes a long time to recover the investment itself, the product needs to be protected. I see a lot of things that look very, very interesting, and unless there's a long enough lifetime to protect that product that is intellectual property, patent life, you're not going to have the opportunity to get that return of investment because you can't protect it unless there's some other novel way of approaching that provides protection for that product, and that's extremely important for us. I think that covers most of the key points here. So again, I think one of the things that really excited me yesterday was Roger's talk, actually, and that in fact there is a wave of innovation that's happening in epilepsy. And actually, from my standpoint, as one of the folks in our company, the leaders in our company that actually decided that epilepsy was not an attractive opportunity, I actually start to see a window opening here, and actually I'm quite excited. So thank you for that.
Well, thank you.
Just a disclosure. So the UCB context, and for those of you, I don't know if the slides are going to be made available to you, that hyperlink will take you to this issue of this Nature review, which we sponsored. And in it, there's some very interesting articles about epilepsy, cutting-edge science, but I want to call your attention to a little bit on the front matter, and there's an email there for you, okay? And that email there is actually a humble and sincere outreach from us in UCB to any of you out here in the community that if you have an idea that you want to share with us, please do so. We don't know what we don't know, and we're certainly open to the notion that there are people out there with good ideas in epilepsy that could help patients. So please feel free to reach out to us. And Henrik, 1-800-HENRIK is our, for those you know, Henrik Littgaard is going to be sort of the point person. You can see the UCB context, and it's a bit of a shameless plug for the company. We are committed to patients with epilepsy, persons with epilepsy, families that deal with it. Keppra is the engine that keeps on giving in 90 countries. Vimpat is doing its work. Brivaracetam is completing its phase 3 development. And Joe D'Souza, who's a colleague of mine, will provide you a little bit of context there as well. We're working with a broad range of companies to work on different aspects of helping patients and persons with families with epilepsy to deal with this disease. And again, we have this broad background in terms of expertise. I think folks have talked about this, so I'm not going to read the slide to you, but the idea here is obviously it's a genericized market with many available drugs and a real problem with the number of drugs that are in early development and in phase 2. If you actually talk to regulators, we're not talking just in the US, but across the globe, they're really not terribly excited about Me Too's. Reformulations are not terribly exciting to them. Regulators are also interested in innovative therapies. And if you talk to payers, they're also not terribly excited about epilepsy. And I'm going to take a moment here and say, I don't agree with the perspective that it's a culture of no. We've sat with these payers, and to the T, they're all interested in providing access to drugs that are meaningful to their patients and to the people who are paying for this. The problem, and I'll get into it a little bit, is they don't understand epilepsy. They have really no good understanding of what the problems. They see a bunch of approved medications, they see a lot of generics, and they think the market is satisfied. And we just actually had a group of these payers, both US and Europe, at a meeting in outside of Brussels. And when you ask them about what do they understand about other parts of epilepsy, the different etiologies, how it's classified, the what's unsatisfied about the market, they have no clue. And so it's easy to say no when you don't understand. And so what I would really challenge this group to do is, they are not your enemy. They really are not your enemy. They just don't understand. And we have a huge opportunity to actually educate them and bring them into the fold to help them understand why access to certain medications may be much, much more important. So the context from a patient perspective, we spend a lot of time talking to patients, interacting with patients and their families to try to figure out what they want. And I don't think it's unique to epilepsy patients. What they want is a cure. Either cure me or prevent me, and if you can't do that, then help me manage the symptoms as well as you can. We understand that getting a diagnosis that's timely and correct is difficult for many patients, okay? Shuttle around between different neurologists. Some syndromes are not that simple to figure out, okay? Getting put on the right drug at the first time is a crapshoot. So some patients get moved around from one drug to the other. We think we have an understanding in terms of rational drug selection, but I would challenge to say that not many physicians really understand how to pick the best drug for the first go around, okay? And if we talk about issues of adherence, one of the major reasons that patients with epilepsy stop taking drug or not compliant is a side effect profile. And there's this notion that somehow poor adherence is an irrational thing. And I keep on pushing about my colleagues that sometimes non-adherence is an absolutely rational decision when you take a trade-off between, let's say, a seizure that happens once a year and not being able to focus on your work, or getting, not able to find your way back home because your attention or memory are impaired, that somehow that trade-off is not necessarily a bad one. And there still, as some of you have noticed in that video that we showed before, there is still substantial stigma. So in terms of the scientific context, I'm preaching to the choir, guys. It's a complex collection of diseases, syndromes, complex genetics. There's interactions between the environment, aging. We know that bimodal distribution. The technology is improving in terms of diagnosis and treatment. There are preclinical models. The iPSCs have everybody excited. It's not just in epilepsy, but I can see and I can hear the vibe here. The notion of being able to take somebody's cells and have a model of the disease in a dish is electrifying, no doubt about it, okay? And from our perspective, although epilepsy is an ancient and historic disease, it still requires specialized treatment and expertise to diagnose it correctly. And in many places in the world, it's not that there is no expertise, there is no access, okay? So when we talk about the issue of emerging of science, so one of the things that we're struggling with is how to actually get the right patients on the right drugs. And what you see here is a whole identification of things that are coming to bear in the area of epilepsy, inflammatory pathways, transcriptional issues, changes in receptor expression, blood-brain barrier issues as well. And those have translated recently into a couple of new areas. And the talk that you heard yesterday from the folks from Sage about refractory status is one example where it's a fairly new population, okay? The idea of super refractory status is fairly new. High mortality, high morbidity, and clearly need for treatments. But it's driven by the fact that there is now new data about the issue of the downregulation of the Gaba receptor. And so if you know that there are now families of compounds that address receptors that are not downregulated, you now have an indicator to a specific patient population for whom a drug is particularly indicated. And if we want to call it precision medicine, let's call it precision medicine. But that's a bit of a driver, is to match a drug with a known pharmacology or specific pharmacology to patient populations in whom that pathology is driven by that particular problem. And as you heard yesterday, there are studies already ongoing in refractory status. Same thing for neuroinflammation. There's emerging evidence of the role of neuroinflammation in temporal lobe epilepsy, neurofibromatosis, NMS. Cytokine profiles are emerging. There are now druggable targets, and there are exploratory studies that were being done, especially with IL-1B and TNF. So if you can now fragment the patient populations, okay, there are markers of inflammation in these particular patients, and you have particular targets that you can go after, now you're segmenting the population, and you have a better match of a patient population with a particular approach. What is not likely to work anymore, okay? We are not likely to develop a drug just because a novel mechanism of action. I think Roy eloquently made the point that differentiation is a problem, and we don't have a marker of why somebody is likely to respond to mechanism A versus mechanism B. So while I applaud, and I can't tell you how happy we are to see people pushing the envelope in new mechanisms, just because it is a new mechanism is not going to be sufficient to actually invest in developing it, okay? Just because it works in every animal model is the same rationale. Just because it works in a broad spectrum, you still haven't handed us in pharma a rationale for differentiation, okay? We talked about the issue of precedent and mechanism of action with little extra. This is particularly difficult because it's, you know, differentiation or superiority when you have a little bit of a tweak in formulation gets very, very difficult to prove. And there's this kind of notion that if you get a drug, you just get it out there, just get it out there. And in a period of a couple of years with 10 to 15,000 patient years of exposure, you'll figure out what makes it special. And I recognize that this has happened more than once. Levetiracetam was a great one, and so was lamotrigine, a drug that went out there with no a priori understanding of what was going to differentiate it. But over time and over use, clinicians found its niche and people really started to understand the value. Again, Roy said it, that model is not likely to work. And so we have to be much more creative and think about how can we actually get time and space to allow a novel mechanism to be used in the wild, okay, to be able to really figure out, does it have a particular space? And here's one of the places where I would say, here's an opportunity to talk to payers, okay? You can argue one of two models. You can say, you know what? Let's get it out there. It's not a matter of an approval. You can get it approved, but you can talk to a regulator and say, you know what, give me 3 to 5 years at a discounted price. Make sure that we have adequate access. And then with some hypothesis-driven studies or observational studies, if with X amount of exposure we find a differentiating signal, then you'll agree to give me a bump in the price to actually get a decent return on investment, okay? You could conversely get pushback and say, we'll give you a premium price for 3 to 5 years, but if by that time you don't find a signal, we're going to discount your price. But either way, and it's not, these are not exhaustive. I sincerely believe that there are models out there that would allow payers and sponsors and patient advocacy organizations to find a middle ground to allow medications with novel mechanisms the time and space to be explored in the real world. And so really what's the hope from UCB's perspective in that you know, mid-sized pharma is that we're hoping that by matching patients with the right drugs that effect sizes will be larger, that it'll be easier and our sample sizes will be smaller because you're actually matching patients to their pathology. Clinical utility will be easier to demonstrate to regulators and payers, to patients, to their families, to their physicians, okay? Benefit risk issues will be improved. So trade-off in terms of better effect and, you know, maybe the side effect profile will be more manageable. You won't have to be struggling every day with differentiation against the multitude of generics. Health economics will be easier to demonstrate. And, you know, we hope that if you have this kind of match in the pathology with a drug, that it'll actually drive patients to be diagnosed earlier and more appropriately because you'll now actually have something that is specifically targeted for them. And so what's missing is really this issue of complementing models of diagnosis with a mechanistic classification. So you know, we talk about in some other areas, whether it's an inflammatory or a metabolic or this or that. So maybe one of the challenges here in the epilepsy community is to figure out, can we start to think about a mechanistic classification? We need to understand how these disorders or syndromes evolve with time and age and comorbidities, okay? And we need to also talk about the fact that there are regional differences in expertise. So treatment of epilepsy in Africa or Latin America, you know, some areas, it's going to be very, very different than it is in New York City or Philadelphia. And I think we have to be cognizant of that. And it's really development, commoditization of clinical trial designs. And this is maybe something that's close to dear to many of the hearts. We need to have clinical trial designs that are commoditized and standardized so we're collecting the same data. Cross-compound comparisons are facilitated. We can train, and this is a lot of what Transcelerate is doing maybe in other areas, is standardization of training of raters, sites, CRFs, so we actually cut the cost of clinical development and actually emphasize the ability to recruit patients faster, better, and cleaner data. So I think there's a lot of work for us to do. And just to kind of conclude for me, plenty of unmet medical need, lots of issues here. Okay, I think there is an informational, educational gap that we need to work with in terms of the payers. Okay, a build it and come approach doesn't work. We have to be able to come up with, hypothesis-driven, scientifically grounded reasons why we think a particular molecule is going to differentiate. Okay, we have to approach this on a global basis, because if you actually look at burden of disease estimates in terms of epilepsy, the real problem is in emerging economies and developing economies. And I think, and this is something I've said, I think it's the best molecule wins, all right? We need to work together to give the best molecule the chance to win out there for the patients that need it best. And I think that's it for me. Thank you.
Thank you very much.
I see everybody's awake. Terrific. So I was just thinking, coming up here, first of all, I want to thank Roger and Jackie for inviting me to participate in this panel. It's nice to have medical device representation amongst this august group. I was just thinking that looking at the titles, or the various titles of the talks, we had, what does Big Pharma think about, investing in an epilepsy product? And then the second topic was, what does Big Pharma think about investing in an epilepsy product? And then medium-sized pharma investing in an epilepsy product. If this meeting were held 10 years ago, we would have had 50 more grades of pharma, all the way down to teeny-weeny pharma. And then, oh, by the way, maybe there's a medical device that might have a piece of action here. And it reminded me of my first AES meeting in December of 2000. And I walked in, and the main purpose was to go through the scientific exhibits and whatever. And there was also, of course, the exhibit floor. And in the exhibit floor, there was absolutely nothing but pharma products. And Cyberonics had this tiny little room off to the side where they were throwing some posters up. And the reason I go through this is that in that period of time, medical devices and the medical device industry really has had much more of an effect and ability to bring therapies to the epilepsy community, and we're very proud to be a part of that. When I think about the device industry and the question that was posed, which was, what does the device industry look for in making an investment in an epilepsy product? If you think about two categories for the device industry, and I'm sure they've never broken themselves up this way, but think of it as either there's an established medical device company, in other words, they're in business, they're actually selling a product or multiple products, or there's the startup community. So if you're an established medical device company, there are two reasons that you would invest in an epilepsy product. The first reason is that you are already in the epilepsy business. You're selling products to the epilepsy community. Therefore, you're selling products to an epileptologist, a neurosurgeon, or a neurologist, again, to treat epilepsy. So what you would look to do under that set of circumstances is to broaden your product line. And we heard an example of that yesterday in the Cyberonics presentation, where the description was made of the new seizure alerting device that they've got. And basically, they're going to go and broaden their product line with that product. So that's an example of that kind of reason why a device company would invest in a product for epilepsy. The second reason would be that the device company has a product or technology that they are selling to another community, another disease community, or series of customers. And that product, with little or no modification, could be utilized to treat epilepsy. And we had another example of that yesterday with the folks from Visualaze, for example. That product was first developed to ablate tumors, brain tumors, and they saw the opportunity. In fact, Joyce Kramer had a big piece from the Epilepsy Therapy Project of encouraging them to apply that technology to epilepsy treatment, and now it's being used, very well, and I'm sure will be used much more broadly. So that's an example of somebody that has an existing technology. We could put other companies in that category, but existing technology to apply. Those are really the only two reasons that an established medical device company is going to invest in epilepsy. If someone is going to invest in epilepsy, that doesn't meet those two criteria, it's going to come out of the startup community. And when you look at what is it that interests venture capital to invest in a company, it becomes not epilepsy specific. It is what makes the best investment for the venture capital community to make. And so how does that decision get made? If they're going to invest in a medical device, there clearly has to be a large unmet clinical need. The company has to have a proprietary or unique approach that is going to solve the problem. Basically, it has to have intellectual property that will protect that approach. There have to be no freedom to operate issues that exist that would cause that type of an obstacle. They have to have the kind of scientific and technical capabilities that can demonstrate to the venture community that, in fact, they've got an opportunity to be successful with this venture. They've got to have a business plan that looks like it makes sense from the vantage point of how much investment is going to be required over what period of time to be able to get what kind of returns, and is this going to be, from a venture capital perspective, the best investment that that firm can make for a potential return to its investors. So they are very disease agnostic when it comes down to that type of decision. It has to be something that will stand on its own to be able to be the firm that gets that kind of investment, or the company rather, that gets that kind of investment as contrasted to everyone else. And then the other kinds of factors that come in there, in addition to the scientific and technical capabilities, what's the management team going to consist of? What's the regulatory path that you have to go through? What's the approximate length of time it'll take to get all of these kinds of things done? And then where's reimbursement? You know, is there going to be an opportunity for these products to get paid for? So when I think about NeuroPace and think about NeuroPace in the list of criteria that I just went through here, I was sitting there thinking that, my God, you know, NeuroPace was absolutely meant to be, because if you think about all the things that had to come together to allow it to exist, it was meant to be. And let me just take you a few down memory lane with that one. So now, 17 years ago, basically, I was approached by an individual that had developed intellectual property for responsive neurostimulation. And the idea was to treat neurologic disorders, epilepsy being the first, but the intellectual property, the patents had already been filed, and the concept was there. And to me, coming out of the cardiac world, I said, my God, you know, this really makes sense. So here you have an episodic disease, you know, the idea of being able to monitor brain activity and then deliver stimulation when necessary. This really makes sense. Let's look a little more. Large unmet clinical need, large market opportunity, order of magnitude 400,000 to 500,000 people in the U.S. that would have partial onset seizures that were not controlled with one or two seizure foci. Basically, the other kinds of criteria that you begin to look at, you say, well, gee, you know, this looks good. Where could we get some initial seed capital? And it just happened that at that point in time, I had just, I'd been a CEO of a cardiac defibrillator company. It had been acquired by St. Jude Medical. I was doing a short stint at Kleiner Perkins as a CEO in residence. I brought it to them. They said, yeah, this sounds like a good idea. So you sort of had that entree that allowed that to occur. And then we said, you know, we've got a lot of interesting stuff here, but we really don't have anybody that knows anything about epilepsy. So what am I going to do about that? Well, okay, we're on Sand Hill Road, which is in Menlo Park. And what's the nearest institution to around here? Gee, that's Stanford. Let me see if I can get a list of people that are, who's running that epilepsy center over there? And I found out it's somebody named Martha Morell. So I call up Martha Morell and say, hey, I've got this idea for something, and would you mind if I came over and had a little conversation with you? And so I'll never forget that day. I walked into her office and told her what we were thinking about doing, and she just looked at me. And I don't know if Marty's out here today. I know she was here earlier, but and she just looked at me, and I'm thinking, my God, what did I say that was so stupid that I'm getting? And all of a sudden, she slaps her hand on the desk and she goes, "We've been treating epilepsy patients for 20 years. Why didn't we think of this?" So, we had one of the most wonderful leaders in the whole community that was right next door, and then interested in working with us. And then it didn't end there, because she had already accepted the job to go to Columbia and run the epilepsy center there. And it turned out that two of the founders of the company, because we had to do proof of principle work, were located right in that upper New Jersey area, and the work that got done was in her lab with general purpose equipment that let us take through the early stages of financing to then be able to have enough of proof of principle to be able and then raise a venture round. And so that's what started our path, and then we developed an external version of our product that we tested in patients with an IDE that were undergoing intracranial monitoring. We then had, in parallel, we were developing the implantable unit. We conducted then 2 clinical trials, a feasibility trial, a pivotal trial, then went through the rest of the regulatory process. And 16 years after our first funding, we actually got approval of our first product and are just beginning to commercialize. And I would submit to you that having those particular events all come together and sequentially allow us to get to this point means that we were meant to be, that this wasn't something that just happened randomly. So with that, I'll just thank you very much for listening to me kind of go down memory lane a little bit, and look forward to participating in the panel.
Thank you very much. I don't see anybody at the microphone, so maybe because we are tight on time, I will ask each of the panelists one question and let them expound, and then we'll call this particular session to a close. So, starting with Santiago, how is it that we can get Pfizer back into the clinical arena of thinking about epilepsy? You have a long history with the companies you've acquired, at least, of being involved in epilepsy. Do you think that we need to adopt the Michael Gold model to get the next drug out?
We are very interested in ion channels. That's an area of strength in our research. So that brought the idea of epilepsy. And maybe the second point is that I am at Pfizer, and I am very interested in epilepsy. So it will always be important for me to be there.
One of the most compelling reasons to have you here, and thank you for coming. Roy, your company is no longer in epilepsy. If we had a differentiated compound, which has been the byword for this morning, if we had a differentiated compound that looked really good, would it entice Janssen to rethink this process?
Yeah, Roger, thank you. It's something I neglected to talk about. And so the perspective I provided was soup to nuts, right? And so do you invest in the discovery work and try to advance this forward, build an entire infrastructure that supports an epilepsy discovery and then early development, late development program. However, I would say every company here remains opportunistic, meaning that if something looks really, really good and attractive, of course there's going to be an interest in bringing that in. And for us, and I'll just set the bar for us where that is, and that relies not at NME, not at first in human, but at proof of concept. And that proof of concept, as I pointed out, was a proof of differentiation. I think right now it would be very hard to convince Janssen to invest in an epilepsy development program that is a commitment to phase 3 development without evidence of proof of differentiation. And so let me just leave it there.
Okay. So, Mike, you've made the most provocative statements about how we might approach payers. I'd like to know since I wouldn't know a payer if one walked in the back door.
That was obvious when you wouldn't pick a checkup last night.
That's correct. Although my wallet is considerably lighter. How do we actually approach it's a two-pronged problem. First of all, we have a ton of drugs stuck in the valley of death, which is pre-IND, okay? And those are, in my opinion, non-differentiable, except from mechanism of action. And usually we just know it's novel. And that's not what you want. I know, like you're shaking your head. But let's assume that we could get some of those through that process and get them out on the marketplace with a novel mechanism of action and good data. How would we negotiate with the payers how would Big Pharma negotiate with the payers to decide whether or not at this stage, because we can't differentiate earlier, we can differentiate in the clinic. That's what we always do. You pointed out levetiracetam, but it's actually true for all the drugs. All the drugs have been differentiated really in the clinic, okay, from phenytoin all the way up to retigabine, which has stubbed its toe, obviously. So how are we going to make that differentiation, and how are we going to find the payers and make that work?
So you pick up the phone and you call Blue Cross and Blue Shield, or you call the VA or Medicare, and they will come talk to you because they're interested in figuring out how they provide better services to their customers. So the payers are not shy. Okay, WellPoint is out there, and if you talk to European agencies, payer agencies, they'll be happy to come talk to you and share their perspective because they're vested in doing the best they can with the budgets that they have. From a sponsor perspective, there are a couple of things that come to mind. One is the notion of sharing savings. Okay, so at GSK, there was what was called the Asheville Project, where they were using Avandia to try to improve the care of diabetes. And they entered into a contracted demonstration project with Asheville that said, we're going to provide the drug for free. And there was a program where there was a control group and intervention group and looked at compliance, adherence, doctor visits, complications, hospitalizations, et cetera, et cetera, et cetera. And the agreement was, or the idea was, that the intervention group would show a lower rate of complications, healthcare utilization costs, and that the sponsor could share in those savings. And there's nothing wrong with that same approach with the treatment for epilepsy. If you have a drug that in routine clinical use really reduces the number of breakthrough seizures, hospital admissions, episodes of status, that has a profile that allows patients with I'm sorry, I keep on referring to them, I'm physicians. They're patients to me. I understand politically it's persons with epilepsy. So if they're able to stay at work longer, do better at school, there's a health economics outcome that can be measured and you can share in that savings. So there are models out there, but they are not simple. They require a long-term view. They require, commitments from multiple parties. And I think patient advocacy organizations are probably one of the most effective, loudest voices to actually get those parties to the table. Okay.
Thank you for bringing that up to us. I think it's a perspective that we hadn't appreciated in that context. And I think the Epilepsy Foundation has to give serious thought to this issue. So the last question of this panel is to Frank Fisher. And Frank, one of the things that it's more scientific question than the business question, but one of the things that I've always been intrigued by from the NeuroPage standpoint is the potential to improve it with time. As you gather data. Could you expand on that? Because I think that that's one of the most exciting aspects of the NeuroPace RMS program.
There's no question, Roger, that that opportunity exists. For those of you that aren't familiar with our system, the device is implanted, actually replaces a little piece of the skull. Leads are placed near the epileptic focus. The brain activity electrical activity is constantly monitored. When detection parameters that are unique for that particular patient are triggered, there are segments of the electrocorticograph that led up to the triggering of the therapy and then a post period that's programmable in terms of length that gets stored and is available on what we call our patient data management system. So this information resides in our system. The physician can access this from any place that he or she has internet connectivity, and the data is there for analysis purposes. And very candidly, when we started this whole thing, we thought we knew a whole lot more than turns out that we actually knew. And so we have been learning during this period of time as well. But as more and more patient data becomes available and more analysis becomes available to us to perform, there's no doubt that we will learn much, much more, and it will be to the benefit of patients, not only in terms of how our system can be optimized for controlling seizures, but what opportunities there may be because of its ambulatory monitoring capability to also feed back information to the physician in terms of drug therapy and the effectiveness of a different drug. For example, we've seen instances where there's been a patient on a particular drug, this is now in the open label period, the physician made a change in that drug, and the proxy for actual seizures is the number of detections. And you can see this just go down in the mud with the response. Clearly that patient has been having a fabulous reaction to that particular drug. Now this is something that normally would take God knows how long, 6 to 12 months just out in the clinic with the patient coming back with their seizure log and the rest of that kind of stuff. But within weeks, you could see that kind of activity. So not only will we learn more about the optimization of our system by itself, but the partnership between the drug therapy and the device therapy and how that works together synergistically, that's a whole world yet to go.
Terrific. Listen, I'm grateful for this extraordinary panel. I greatly regret that we have to stop because of time, but for all of you, my colleagues are probably going to be here for at least a few more hours, so please avail yourselves of these phenomenal experts in their field and ask them questions individually. Thank you very much for coming.